Welcome back, students, to gene interactions, and we are continuing with the allelic interactions. We had mentioned about the complete dominance, incomplete dominance, as well as the codominance. And right now, we are going to explore the cumulative genes of the multiple genes, as well as the pleiotropism. What is a polygene or a multiple gene? This name was mainly been coined by Mather K, and you can see that sometimes the characteristics of some offsprings, instead of being sharply defined or sharply marked off from one another, they will show some continuous variations. And this is known as a type of quantitative inheritance. And you can see that if these characters, these quantitative characters, are being governed by numerous duplicative, cumulative, as well as non-codominant genes or non-dominant genes, such genes are called the polygenic genes or the polygenes. And they mainly differ from the classical Mendelian pattern in that the entire range of variation is being covered in a graded sequence from one parental extreme to the other. I guess you are getting confused. What is this polygenes or multiple genes? Now consider a case in which the two extremes of a particular character are not being expressed, but in between the different gradations of that is being found. Now consider you have a Negro getting married to or getting married to a white. Okay, so the offsprings which might result might show different gradations of either of the parental traits. So the human height as well as the skin color is an example of a polygenic trait. That is, it has been influenced by more than one, two or three genes, and it shows gradations between different type of variations or different variations of these two extreme genes can be found. Or if you go to consider the coat color in the case of cattle, or the size in the chicken, or the egg laying in the case of chicken, these are all examples of polygenes which will code for quantitative characters. You can see that these polygenic traits, they are usually controlled by more than one particular gene. And instead, you can see that several genes will contribute to the final phenotype of a particular trait. So the color of the human skin itself is being controlled by, rather than one single gene, many genes are being contributed by or contributing to that particular character. And each gene will play a small role in the expression of that particular trait. And this will allow for a trait to vary greatly from person to person. Now, height, as we mentioned earlier, it is a polygenic trait, and that is the reason why some will be extremely tall, or some will be short, or sometimes the offsprings will be in between that, or anything in between, or highly variant. So that is an example of a polygenic trait. So this is an example of a gene interaction. As we can see, you have multiple genes are being found to be involved over here. Now, another example of allelic interaction which we have to talk about is pleiotropism. Now, what is pleiotropy? Pleiotropy is just the reverse of polygenic trait. Now, you just look here. In case of polygenic traits, various genes are being contributing to one particular trait. You just take the reverse of it. That is what? One particular gene, okay, one particular gene is affecting various traits. That is the case of pleiotropism. So polygenic genes or multiple genes are just the opposite of, you can just reverse these two characters and then you get what is polygenic trait as well as pleiotropy. So if one particular gene will affect various traits or affect multiple traits, that is referred to as pleiotropy. And if a mutation in that particular gene, now if this particular gene is responsible for 4 different traits, That is a case of pleiotropy. And if this gene gets mutated, that mutation will affect the expression of all these four traits. And that has been found to be a very severe case of mutation. Now, if you go to consider sickle cell anemia, you have a in the case of sickle cell anemia, here also there is a mutation of one particular gene is being responsible, where glutamic acid is replaced by valine over here, and you will get a sickle cell shape of a hemoglobin is being formed over in sickle cell anemia. And you can see that this beta globulin gene mutation would affect different traits. Now, it could result in the formation of blindness. It would affect in liver failure as well as in heart attack. Like that, different types of traits get affected because of the because all of these are being coded by the single gene of beta globulin. And a mutation in that which results in sickle cell anemia or the sickle cell shaped of blood cells, okay, RBC cells, would also affect various other functions as we mentioned here. The rapid breakdown of the red blood cell will cause anemia, leading to the increased lethargy and a higher risk of infection. And the clotting of the sickle cells in the vessels can cause heart attacks and brain damage, as well as in the case of strokes and paralysis. And the accumulated blood cells in a specific organ could also lead to the loss of the function. Now, for example, liver or splenic failure or kidney damage, etc. This all can happen as a result of the mutation which is happening in one particular gene. Now, another example which we have is phenylketonuria. Now, in the case of a normal individual, phenylalanine is an amino acid. That amino acid will be converted into another amino acid called tyrosine, okay, using an enzyme called the phenylalanine hydroxylase. Now, so in such people, now if phenylalanine is being found to be in a high concentration, what happens? That is not good for the blood. When it accumulates in the blood, what happens? It is being found to be toxic to the central nervous system, and it will damage the brain and cause mental retardation. So, a person who's having a phenylketonuria, what happens? They have a autosome. It is an autosomal recessive metabolic disorder, and a gene present on the chromosome 12 is being found to be affected. And as a result, what happens? Less or even null amount of phenylalanine hydroxylase is being formed. And as a result, what happens? The phenylalanine will not be converted much into tyrosine. And phenylalanine in the cell will be found to be what? Accumulating. And so, by the damage which was being caused on one particular gene on chromosome 12, what happens? Phenylalanine is not being converted into tyrosine. And the presence of phenylalanine in the central nervous system, as I told you earlier, it will cause the brain and the mental retardation. At the same time, you can see that for the production of melanin, okay, melanin is a pigment which has been found on the skin. You need tyrosine. So what happens? When this mutation happens, tyrosine is also not being formed. And as a result, it will reduce the skin and the hair pigment formation also. So the mutation in one particular gene, which is being located on the chromosome 12, is resulting in mental retardation and reduced skin and the hair pigmentation. So this is another case of pleiotropy, as you all know. I hope you got a better understanding of what is pleiotropy and what is multiple gene. In the case of polygenic traits, what happens? Various genes are contributing to one particular trait, and that is in the case of the human color. Human skin color is an example. But in the case of pleiotropy, what? It is just a reverse. One gene is affecting various other traits, and hence a mutation of that particular gene will cause a mutation in various other in all these multiple traits which are being coded by them. And examples which we have is about sickle cell anemia as well as the phenylketonuria. I hope this is clear to you. With this, we come to an end of the allelic gene interactions. Thank you for now.